From the College by the Lake, Coeur d'Alene, Idaho. Local, regional, national, and international guests discussing the issues and topics affecting the way you live are on Forum, the North Idaho College Public Forum, with your host and moderator, political scientist Tony Stewart.
All the news media has been recently discussing a major breakthrough in the field of science, and I know all of our viewers that you have been following this program or subject, and that has to do with DNA research, or we call it genetic engineering, and in this case, genetic engineering with the process of cloning. A Scottish scientist recently had a breakthrough in which a cell was taken from a sheep and through implanting it in a egg of another sheep came up with a single somatic cell cloning process. We wanted to, particularly on this program, talk about from a scientific perspective what that means, and also some of the results that might happen in the future because of this particular development. I am so pleased to welcome to our program three members of the faculty at North Idaho College who are certainly very well qualified to discuss this subject, and we're so pleased they took time to be with us today. And it's always a special privilege on this program to have my colleagues here to join in discussing an issue. I first of all welcome to the program David Cunnington. He holds a bachelor's degree in zoology from Eastern Washington University. David, welcome to our program. We're delighted to have you here. And next to him is Peter Zao, who is also on our faculty. He holds a master's degree in biology from University of California. And I should back up and say that, apologize to David, he also holds a master's degree, not just the bachelor's degree. And then next to Peter is Mike Herod, who also holds a master's degree in biology from Eastern Washington University. Gentlemen, welcome to all of you, and we're pleased you've taken the time, as I indicated to David. And I'm always pleased to have Janelle Burke, who is our regular panelist, to join in the program to discuss what is a very major development issue. On the format today, Peter, I shall commence, and I want to start with you. I know a lot of the viewers are in the same category I am, not understanding this process, and would you talk about how this particular scientist took a somatic cell from a sheep and what that process and what the nucleus was and how that came into the results that we now have?
Well, we basically want to think of a cell in a developing organism such as a human being or any animal when we have the union of an egg cell with a sperm cell. Then we have a complete set of blueprints to generate another organism. And when this new process was put together, this is something that's been attempted for a lot of years. We're concerned with two primary pieces of an embryo. First of all, the egg itself. The egg contains a membrane that contains within it all of the fluids, the enzymes, the proteins that are necessary to read the blueprint and to start building this new organism. Second, we need the blueprint itself, which in human beings is a set of 23 pairs of chromosomes, each containing thousands of genes that code for individual proteins that make up a person. Those proteins can be either structural, building things, or functional, in the sense of causing chemical reactions to occur. Now, what makes this whole process so unique is that the DNA from an egg cell in one sheep was removed, and the DNA from a somatic or a fully developed cell in another sheep was taken out and put into the original egg cell. So now we have an egg cell that has a totally foreign piece of DNA in it. And the blueprint now codes for the sheep that we took the DNA from. And we want to think of the genetic material, the DNA, really as a basic set of blueprints. And just like with the blueprints in a home, if we read those blueprints, we have a competent person to build that home, then we will have something come out of it that is structurally what we expect it to be. And if we took that to another individual, another builder, we would expect that home to at least reasonably resemble that first one because the blueprints are exactly what has been specified here. And so in essence then, from the sheep that we have taken the DNA from, we will now have an exact duplicate of that original sheep. And it will be, there can be some variability in it because we always get something that changes during the development. For example, if the mother, something happens to the mother chemically or biologically, then we can have variation in that in that offspring. However, genetically, it will be absolutely identical to the original.
Mike, I want to follow up on that process. We're saying that from Peter's conversation that it is as close as you can get to being identical. You indicated before the program to me that without the cloning process that it's 50-50 as far as DNA from the two animals, the male and the female, and now we're looking at different. Explain to us, too, that there are still some real complications in how you put this together on the cycle and so forth, that it just can't be done any one day, can it? But just taking a cell and doing it without understanding that process.
Right. I think when you take a cell that is specialized, such as what they did in the sheep, that cell is following a normal cell cycle. And if you were to take that nucleus that is caught in that cell cycle and then put it in an egg that is in a slightly different cycle time, unless those cycles are somewhat synchronized, the nucleus from the specialized cell will be on its way to doing something that the egg wasn't on its way to doing. So this may be the whole kind of crux or key to this is being able to figure out how to synchronize this transporting of a nucleus into the cytoplasm and the membrane of another cell and synchronize the cycles.
Is that rather easy to do, or is it pretty complicated to know what the cycle is, and is it by luck, or can you determine that from some scientific measurement?
Well, I'm sure it's determinable. I don't think as yet it's been determined. I think that was one of the discoveries when he was able to clone the sheep that kind of set people back and says, now we know it can be done. Let's figure out how it was done, why it worked in the first place. Because he tried, I think David told me earlier, he tried about 273 times to do this. And on the 273rd time, he was able to catch the right cycle. Now, whether that was purely by accident or he had figured something out, we're going to have to see what his notes say when he publishes.
Yes, I introduced all of you in the program. All of you hold graduate degrees, two of you in biology and David in zoology. And you certainly have a background that's valuable to us. And following up on that, David, what is your opinion as a zoologist on being able to be rather accurate in determining, or will we continue to do it like Russian roulette? You make the combination by simply trying.
I'm not all that versed on exactly how his process was. I haven't read anything where he had published any recipe, so to speak, exactly how he went about that. One thing that we know in science, or one thing we hold hopefully true in science, is that everything can be figured out given enough time and enough energy put towards something. So I'm under the impression that it's a matter of time, a matter of money to get the research going to actually figure out or make it a 100% thing. If you want to clone something, it can be done. We just do it.
Before I go back to Janelle, and she'll have a series of questions, I just have one more filling in this particular sequence. I was talking with Peter in particular before the show and was really interested, and David, I'll let you deal with this, but that even though they genetically have this same formula, that as two animals are growing up, or one's already grown, the other one's growing up, there's things that can be different, both even physically and certainly if you get into other issues, the diet and all, when we say identical, there's still differences, isn't there?
Absolutely. We don't know exactly the full extent that the environment plays on an individual's genome, but we do know that it does play a significant role on their genome, and that can be seen in human identical twins. They're not absolutely identical. They may share the exact same genotype, but when you look, or the exact same genes, but when you look at them as an individuals, you can always tell them apart. My father-in-law is Don Burke is a twin with his brother, and they, I mean, on the outwards appearance, very, very much to look alike, but when you get to know them, there are a lot of differences. And so the environment does play a large role as far as the diet of the individual, possibly even any type of maybe pathogen or any other environmental stimulus that they have received that the other one did not receive could in fact change their outward appearance, or at least their genetic
Peter,
manifestation.
Would you like to add to that in any way?
Well, that's David has pretty much covered what it is. The idea we need to keep in mind is that no two people's environments, even if they live together, is exactly the same. It's just like when in a family we get an individual that has the flu, that doesn't necessarily mean that we all will. Each one of our physiological systems is going to treat that organism in a different way. And the bottom line here is that even if we have absolutely genetically identical individuals, that does not mean that they will respond in exactly the same way. And the longer that their lifetimes are, the more they will physiologically tend to diverge away from each other.
Janelle Burke.
We're talking about a very new subject, and I know there are lots of there's lots of interest out there in this subject, but there are also a lot of misconceptions. And so I want to ask a series of questions that are kind of the sort of thing where these are just interest questions. They're answering some of the unknowns yet about this new subject. Let's start with you, Mike. And as I understand, you are a botanist. Mm-hmm. And is this a new thing? How do you clone plants?
In plants, little different than what they did here. It's not as it's kind of become a standard treatment in the lab to do a plant cloning lab, where the students get a plate with regulated hormones within the medium of the plate itself, and you take a cell from the plant, put it back in the hormone, and then it will follow a cycle where it will form somewhat of an embryonic stage, and then you transfer that into a different type of medium with different environmental factors coming into play. There are different hormones, different nutrients, and it will differentiate into a new plant. So it is the same in that you're taking a cell back to the embryonic stage and starting over again. It doesn't seem to have the social implications that this does.
So what you're saying is that we've been cloning plants for a while.
Yes, we've been cloning plants for a while, and we've also cloned animals before.
And what kinds?
In the 50s, they were able to clone frogs, where they would do the exact same process that Pete described to you. The only difference that we see in that process from the 50s and now is they were using an early embryonic cell that had not become specialized. So it's really similar to identical twins, where an egg divides, or the zygote goes through its first division and then they separate. So we actually have two different zygotes with identical genetic material. They did the same thing. They just took one of those early divisions, did the same process of taking the nucleus and putting it in an egg that had been enucleated, or the nucleus was taken out, and got twins. So
that's what they were doing with the frog.
Right.
But this is a little different process now, right, that we've gone one step further, in other words, with the new process.
Like in all science, we really just answered one more question of, can this be done? And I'm sure that quite a few people were working on this.
Now, is the process, Pete, the same for every animal, or does it get more complicated as you rise to a higher species?
It undoubtedly will be different for each organism. The frog is not nearly as difficult. The lower you go down in species complexity, the more, I should say, the less complicated the blueprint is, the DNA. And the higher you go, the more difficult it is to turn on the genes at the appropriate time. I think what Mike said is a key point here. In the 50s, that was done with embryonic tissues. Those tissues were not determined yet, and now what's exciting about this process is that we have taken an adult cell, one that is already determined to be a cell from an udder, and from that udder cell, we have now created an entire new organism. It's been known for a long time that all of our body cells, virtually all of them, contain the entire blueprint for the entire body, so theoretically, cloning was always possible. The key point has been, how do we turn on those genes that were previously turned off? And that's what this is a breakthrough in.
So what you can get now is you can see I've got a satisfactory product here that I want to clone. Before, you didn't know what you were going to get.
Exactly.
You had to live with what it was that was produced in the future. But now you know I've got something here that's satisfactory. Now, when this happens, it grows through all of its stages. Is that correct?
That's correct.
And so it grows just like it starts with the baby, and it's born. It starts with the baby.
Exactly the same.
And it's going to grow up. Now, in terms of how it's going to look, is it going to look exactly the same, David? Is it going to look just like the one that it matches? Is it going to have the same markings? For example, in the sheep, is it going to have the black face, look exactly like the original?
You would expect that to be true, since the outward appearance of an organism is manifested by its genes. And so if there is the exact same genotype in each organism, then they should look very much so exactly alike. Although I would assume also that the environmental conditions do play a role in this case also. So if they were raised within the same boundaries, I would say that their outwardly appearance to us, because a sheep is a sheep is a sheep, for the most part to us, they would look absolutely identical. And in fact, they really should be, because they do contain the exact same genes which their looks came from.
So we should be able to identify a clone by its markings. That is, at least in animals. The black and white cow and the particular spots on the Holstein are going to be in the same spot?
That seems reasonable, that you should be able to say that cow, because if you've ever been to a field that has a bunch of Holsteins in it, you may not be able to tell them apart. But if you spend a lot of time with them, you would be able to tell them apart by those markings. And so if you were to spend a lot of time in a field that had a lot of cows, that all were black and white spotted, and there was a clone out there, sooner or later you would figure out which one it was by the markings, because it looked exactly like another one.
So from that, Peter, I can assume that if we can then go to the survival of the fittest, and we can begin to clone for those qualities that we really want to have.
Theoretically, that is possible. However, when an organism goes through its developmental stages in utero, the environment of the mother also does play a great role. For example, if you take the extreme of the thalidomide problem a number of decades ago, those mothers were taking that as an anti-nauseant, and those children were then born without arms or legs. Now, their blueprint was fine, but the environment that the mother had been put into had changed them significantly. So then, even if we do take a section of DNA out of an adult animal now and place it into another egg and then grow that egg, the environment of that mother does play a large role in the outcome of the individual.
I want to take this a step further with Peter, because in answering Janelle's very fine questions, you were indicating that as you move up in the animal kingdom, that the combinations become more complex. If you move from, say, a sheep and there's an attempt to clone humans, which I know is a very, very controversial issue, and we said, and I should say to the viewers, we didn't call you here today to talk about the ethics or the public policy. That's in another arena, and others will decide that. We ask you as scientists to talk about the process. But would that be more complicated and less likely to succeed from a scientific perspective than what we have just observed in sheep?
Well, this researcher has indicated that it would be very difficult to clone a human, if not impossible. And that is a reasonable statement for him to make. The genetic composition of a human must be very complex, given our complexity. Without really knowing, though, it is very difficult to say. This is groundbreaking research, and something like this has not been accomplished before, in essence, turning on DNA that was previously determined. And so for that's something that's coming with the future.
Peter, we always have an advantage of talking to you before we go on the air, and we learn some things that we can use on the program. I was also taken by the fact that you said, as we move forward and talk about what effects this may have in the future, would you share with the viewers what you discussed with Janelle and I about if we were to clone cows, for example, and we got to the point where all cows had an identical appearance or DNA, what that could be as a consequence to society down the road?
There could be some danger in there, given that you do have a perfect cloning procedure and you do create absolutely identical genetic individuals, and it is created generation after generation on a continuing basis. Then soon you have, in essence, removed all the genetic variability out of the population. And genetic variability is what allows the population as a whole to survive. If, for example, you have an individual that is susceptible to some type of a disease that they have not come into contact with before, if if that is a terminal problem, then if all the population is identical, then they will all succumb to that.
If that is triggered and becomes part of the problem for
that. It could be something that wipes out an entire population.
Well, I hear you saying, though, as long as we have variants in the genetics, then some groups would survive while others would be destroyed by the problem of the disease.
Exactly right.
I want to ask both Mike and David to proceed with part of what I said would be on the program today, and that is, now that we look at this rather remarkable development, what are some of the other things that, either positive or negative, that you see that could happen in the future? Because, Mike, I'll start with you, as you talked about the frog and now moving to the sheep. Certainly when scientists do these kind of things, they learn so much that there is a next step. And so what do you anticipate might happen from this development?
As far as the kind of agriculture end of it, talking about the sheep and the cattle and having identical individuals, as long as the biodiversity is maintained, it'll probably be heavily regulated to maintain the biodiversity, but as long as it's maintained, it would be quite a financial gain for, say, the rancher to have a herd of cattle that could produce, maybe, for example, high marbled quality meat within the same time period, maybe even a shortened time period, and have all his cows come into being ready to go to market at the same time period, instead of having, say, weaker or stronger cows and trying to adjust that. Same on the milk products, having a cow that will produce high quality milk. If you cloned those cows and had a herd that produced high quality milk, that would be of a financial gain also. We were talking a little bit about the medical industry and some of the products we use from animals, and we want those proteins to be as close to ours as we can get. So if we have an individual, as in a sheep, because we use a lot of sheep milk proteins, if we have an individual sheep who creates a protein that matches ours, fairly close, it won't be identical, but fairly close, we could clone that sheep. But even one step further, if we can genetically manipulate the DNA in vitro from that sheep and then clone, we could have these animals producing identical proteins to ours and using them in medicine.
To try to avoid the concern that Peter has raised, in the cloning process in the future of animals, if that's the path that is taken, could you clone different animals in different categories, and therefore if one group got wiped out by a certain disease, you'd still have another genetic formula?
Well, yeah, I think we've learned our lesson with the monoculturing of the corn and the potatoes and the tomatoes, where we see these blights that come in and wipe out a whole section of our country's, say, corn crop, because they're so genetically identical. They couldn't fight the disease. There was no natural selection to take place in there. If we take that into consideration and keep proper records and understand that we have to maintain the diversity, what's good today is not necessarily good tomorrow. So if we continue to try and satisfy our needs for today and not take into consideration what we're going to need tomorrow, we may end up, as Pete said, hurting ourselves, and it could be pretty dramatic.
And David, would you share with us some of your thoughts and ideas of what else might develop in the future because of this particular cloning process?
Well, I would like to stop for a second and let you know, or let the viewers know that the cloning process is not a man thing. It's a natural process. Lower organisms do it all the time. Any means of asexual reproduction is a means of cloning yourself. You're getting an organism that is exactly like the original. So you have a flatworm, and it divides by fission. Each worm grows into another worm, and they share the exact same genetic material. The problem with that, and over evolutionary time we've solved that with sexual reproduction, but is a lack of genetic diversity, and then these things that these gentlemen have talked about come into play. Any type of environmental change that could kill the parent, so to speak, organism would also take any of its clones with it. And so I guess what I wanted to make sure that everybody understood was this isn't a new thing by any means. It was around long before humans were around, as far as evolutionary history goes. And so it's probably something not to be afraid of, but something that maybe we need to definitely keep our eyes on.
Okay. Janelle Burke.
David, I want to ask you about, for example, the racehorse. We have champion racehorses. What happens if we clone that racehorse? Are races going to be dull?
Well, as far as I'm concerned, and this is definitely my own personal opinion, this would be a misuse of cloning because it's good for one person or one group of people rather than all of mankind. This is more of, if I were to regulate this, it would be something that was good for mankind as opposed to certain individuals. Like as Mike said, if you could maybe help to support the world with beef production and high quality grain and whatnot in this fashion, then that makes it a pretty good thing. But cloning a racehorse, you know, in the racing, is that a sporting means of getting a better racehorse? I tend not to think that's very good. But then once again, that's my opinion. I'm sure there's racehorse owners that think that's a great way of getting a very good chance of getting a winner. You know, because of course there's a lot of other training and whatnot that goes into a racehorse that actually makes it a winner. It's not just the genetics, although the genetics do help considerably.
We're getting fairly close to the end of our time, and if you can believe that, this has gone so quickly. But I want to give all of you an opportunity to say, if you can think of anything that we've missed today, particularly in view of what are the advantages or the disadvantages of the cloning process, and what should people be watching for now as the media begins to cover these events?
Well, I think this may be kind of our first chance you know, as this generation to actually get involved and make a decision based on morals and ethics, which we can only make as if we keep ourselves informed as this progresses along. So many people become discouraged by reading something they don't understand and feel that their opinion wouldn't be valid in determining should we breed race horses, or you know, should we clone for new medicines, or you know, and if what I'm trying to suggest is if the opportunity arises for your input, that you should be informed, because there's a lot of good that can come from this, but there's also probably some bad that can come from this. And as long as we pay attention and keep current, you know, our own opinions will be worth putting into the pot when it comes to should we do it or not.
On that note, I have to bring the program to conclusion. Because of Janelle's question and for many other reasons, we should have you back in the future. You've been an excellent guest, and we'd like to do that and talk about this and some other things. I would like to take advantage of your expertise and having you here on campus. And gentlemen, thank you so much for being with us. Ladies and gentlemen, our issue has been cloning and what's happened recently in that field, and I'd like to invite you to be with us again next week at this same time when we will discuss yet another interesting issue. Until then, please have a good week. I am Tony Stewart.
The North Idaho College Public Forum was videotaped live from the studios of Instructional Technology on the campus of North Idaho College for viewing at this more appropriate time. We invite you to join us again next week for another all-new edition of the North Idaho College Public Forum on this public television station.